fantastic to see you here tonight. It's fantastic to have an event in person. So a very, very warm welcome into this beautiful, absolutely gorgeous space. And we're eternally grateful to Katja, who's sitting back there. Katja, can you raise your hand a little bit? Who's the director here of 1014, and she personally opened the door to us tonight. So my name is Catherine Paula. I'm the program coordinator at the German Center for Research and Innovation. In German, we call it DWIH. Can we practice DWIH, all of you? DWIH. So you'll never forget that again. Great, fantastic. And we're very, very happy and delighted to co-sponsor tonight's Leibniz lecture, which is a very special one with the DFG, a long-standing partner of ours. And I'm also speaking for Ben Grisch, our director of the German Center for Research and Innovation, when I say thank you, Georg, the director of the DFG, for having us here with you tonight. A little bit of housekeeping. So we'll have a few words of welcome. We have Yasmin Pamuk, she's the head of the Culture and Science section at the Consulate General, and then we have Georg, I just said he's the director of the DFG, another German abbreviation here in North America, and then we have Matthias, welcome, who's going to be our moderator tonight, and then Professor Prinz, who is just passing through and happens to be here with a fantastic title, Watchdogs of the Brain, and he's going to enlighten us what that means. So with that, I'm stepping aside and open the floor, leave the floor to Yasmin, who's going to do a proper introduction to tonight. Thank you very much and welcome again.
Meine sehr verehrten Gäste, dear partners and friends, lieber Georg Bechtold, liebe Katja Wiesbrock, lieber Ben Grisch, distinguished professors Matthias Haury and Marco Prinz, ladies and gentlemen. On behalf of the German Consulate General New York, I welcome you to the Leibniz lecture, Watchdogs of the Brain, How Local Immune Cells Regulate Health and Disease. Humans have walked on the moon and sent probes all over the solar system. Nonetheless, we still do not fully understand how our brain works, how its structure, chemicals, and connectivity interact to produce our thoughts and behaviors, and what exactly happens if our brain cells degenerate and do not perform their proper functions anymore. Tonight, Dr. Matthias Haury and Professor Marco Prinz will remedy our ignorance by sharing their valuable insights about the origin, fate, and function of so-called microglia. I imagine them as some kind of huge cookie monsters that munch their way through, quote, invading pathogens.
Cookie monsters, that's good.
The namesake of the prize and lecture, Gottfried Wilhelm Leibniz, would certainly have applauded tonight's Leibniz lecture. As panpsychist, Leibniz believed that everything, including plants and inanimate objects, had a mind, or something analogous to a mind. More specifically, he believed that in all things there were simple, immaterially mind-like substances that perceived the world around them. Leibniz called these mind-like substances monads. While all monads had perceptions, so Leibniz, however, only some of them were conscious and perceived their inner and selves, and had some sense of their selves. Some of them only possessed the sensation of consciousness. Even fewer monads were capable of self-consciousness and rational perception. But what happens if we slowly lose this sense of self? As human beings, we need to know that we are and who we are. We need to be proud of our abilities and feel respected by other persons. Today's Leibniz lecture will explain why and how these experiences are inextricably linked to the above-mentioned cookie monsters. That is, what crucial role microglia cells play in a wide range of neurological diseases like Alzheimer's and multiple sclerosis, or better, how to prevent them. On the assumed interconnectedness of body, that is brain, body and mind, that is brain, however, Leibniz might have contradicted Professor Haury and Professor Prinz and modern neuroscience vehemently. In several of his writings, he argued that purely material things such as brains or machines, to what he compared brains, could not possibly think or perceive or cause physical action. Instead, Leibniz put forward his doctrine of pre-established harmony, saying that mind and body, without any direct causal links, naturally corresponded to one another. More specifically, Leibniz believed that God created our brains and bodies in such perfect harmony that they remained perfectly synchronized without any causal links or adjustments, let aside macrophages or microglia. Let us think this through. God knew, before he created our body, my body, that I would decide to speak here tonight, and hence made my body in such way that I would, in virtue of the nature of my body, take this microphone, open my mouth, and utter these words, while my brain made the very decision which words to use. Leibniz contends that in this way, God ensured that the state of mind and body corresponded in a perfect harmony, from his lips to God's ears. Ladies and gentlemen, cutting-edge research is highly depending on international networks and exchange. Germany, therefore, attaches crucial importance to events such as this Leibniz lecture, a marvelous format organized by the Deutsche Forschungsgemeinschaft that honors Leibniz Prize laureates and brings them into dialogue with research communities as well as a broader public all over the world. This form of science diplomacy has long been a tool to foster and strengthen not only international relations as such, but specifically transatlantic relations between Germany and the United States. Our relations are shaped by common values as well as shared interests and the commitment to excellence in research and, of course, our appreciation of academic freedom. I very much look forward to this year's Leibniz lecture here at this wonderful place and wish us all an insightful and inspiring event with Dr. Matthias Haury and Professor Marco Prinz explaining Cookie Monsters to us.
Yeah, ladies and gentlemen, this was the philosophical part of today's evening, and now some statistics will follow. But let me first start to explain what DFG means. DFG. This is a German Research Foundation, and it's quite similar to the National Science Foundation, you may all know. However, we don't fund only natural sciences, but humanities as well. And our budget is about 3.5 billion euros per year, which corresponds to, I think, $4 billion. And part of our budget, the largest part of our budget goes into unsolicited proposals, research proposals, but I think 1.5% of it goes into prizes. And the most important prize of DFG is the so-called Leibniz Prize. And this Leibniz Prize is given to about 10 laureates each year, and everybody of them gets 2.5 million euros, which corresponds to $3 million, and has 7 years to spend it, but to spend it only for research. Yeah, so it's less money than the Nobel Prize. However, with a Nobel Prize, you can buy yourself a Porsche or a Ferrari or whatever. But as a hardcore scientist, I think you don't need a car, and you have a bike. Research. So yeah, the statistical part. Need my glasses. There are until now 923 Nobel laureates, and 10 of them have been Leibniz awardees before. So this is a percentage of 1%. Yeah, 1%. And today we had 415 Leibniz awardees in total, and 10 of them, as I just said, are Nobel awardees as well. So this gives a percentage of 2.2. So the probability that you listen now to a future Nobel laureate is 2.2%. Yeah, and with that probability, I hand over to Dr. Haury and to introduce Dr. Prinz, please. Thanks a lot.
Thank you and good evening, and thank you to all the partners that have made this evening possible. My name is Haury. I'm the Chief Operating Officer of the Max Planck Florida Institute. Most of you probably don't know that there's a Max Planck Institute in the United States. It's the only one, and it's located in Jupiter, Florida, not Jupiter the star, but Jupiter in West Palm Beach, very close to where our former president is actually located these days. So if anybody is interested, it's close to the beach, very nice. And we do groundbreaking neuroscience, which brings me here today because I think Dr. Prinz is professor at the University of Freiburg, is also doing groundbreaking neuroscience, actually more than groundbreaking neuroscience, neuroimmunology, which is an area of research that becomes more and more interesting. I myself actually am also immunologist because I studied immunology at the University of Freiburg long time ago, so that's how the paths cross, not in person, but at least virtually. And I'm really, really pleased to be able to assist to this lecture today and to introduce Marco for, first of all, and congratulate him to the Leibniz Prize, because I think this is something he didn't have a chance to deliver a lecture in person, so this is, at least in a small format, one opportunity for that. Marco studied in Berlin and got his MD degree at the Humboldt University, or formerly called Charité.
It's still called Charité.
Still called Charité.
Yeah, it's a medical campus.
You know more about the German universities than I do. And then went for his residency to the University Hospital of Zurich in Switzerland, and did really groundbreaking research there. I'm not going to unveil all the details because I think he will probably talk about some of those results. Before then being nominated as a professor in neuropathology in the University of Freiburg, where he's working since, and he's really trying to do research which goes a little bit outside the box and maybe in line with Leibniz, trying to kind of figure out how the body actually interacts with the brain and how the immune system manages to go around or about with the blood-brain barrier. So I think I'm really curious to see what you're going to tell us tonight, and then we can discuss and get further insights later on. So without further ado, Marco, thank you very much.
Super, okay. Okay, thanks a lot for these very nice words. A warm welcome. Special thanks for our organizers, Alte Vogt and Herr Bechtold. It's a great pleasure to be here. It's an honor for me to present our work. And, you know, our main interest is the brain, you know, and the brain is a very complex organ, basically. And what you already see here is, for us, the most interesting cell. It's an immune cell inside the brain. And I will come in a minute to explain what this cell is. And actually, I brought 2 slides to introduce who we are and what we are doing. This is a round of us, MDs, sitting around the microscope. And we are part of the University Hospital. And as such, we have diagnostic duties. What you appreciate here are brain slices, brain specimen from patients that get operated. And as our diagnostic duty, we have to diagnose here brain tumors, metastases, any kind of brain diseases from living patients, basically. Several thousand specimens we get every year and make the diagnosis of brain tumor, for example. However, this is just the MD sitting around, but the whole institute are much, much more people. These are technicians, secretaries, and researchers. And all these people here, part of the Neurocenter, are dedicated to do either diagnostic or research. And some of our diagnostics, it's also on the dead people, brain autopsies here, where we cut the brains. But the majority, as I said, we have specimen biopsies from the brain from several thousand patients per year. So, well, I would like to introduce the brain a little bit, so that you also take something home. And if you look at the history, who was first interested in the brain, and it was already 3,000 years before, where the Egyptians were interested in the brain, you know. For these people, different organs were very important, as they put it here in the canopes. They removed the liver here inside, the spleen, the kidney, and the heart. However, the brain was not put inside because for the Egyptians, the brain was not important. They just remove it through the nose because if you wait for some days, the brain gets liquid and it smells very badly, basically. And that's why they just removed the brain and it didn't appreciate that the brain is so much important, 3,000 years back. So, and if you look in the 19th century, colleagues of mine, neuropathologists, were quite broad scientists or medical doctors. And one of these people here is quite famous. It was Alois Alzheimer. And at this time, MDs still had patients, or neuropathologists still had patients. And this is a brain from Auguste Deter, who was his patient, and where he did biopsy later on and diagnosed the brain. What you appreciate here is the temporal lobe, and it's very small. That means there's already an atrophy, degeneration of the brain, and this disease was later called after Alois Alzheimer, first description. So, and this is also a quite famous neuropathologist. Some of you might recognize him. Like a Danny with a cigar here. Who is it?
Sigmund.
Sigmund, exactly, Sigmund Freud. He did his habilitation in Vienna on the spinal cord. He has the spinal cord changes, the spinal cord, and he was for many years dedicated to neuropathology. And afterwards, he moved on and left physically the brain. So, and but nowadays the neuropathologists look a little bit different, as you see in my example. Well, if we look inside the brain, I would like to recapitulate what cells we have in the brain. Just a little bit quiz. Cell number 1. Any suggestion what cell type this is? It's the biggest cell type by far, but not the most frequent one. No. Astrocyte would be number 5. Yeah. Astrocytes, yeah. But the biggest cell in the brain is the neuron. Yeah, it's a neuron. Astrocyte would be number 5. Number 2, what cell type in the brain is number 2? Yeah, well, it's a special glial cell, it's an oligodendrocyte, in close contact to the axon, here, neuron, axon. Astrocyte, it's a brain vessel. And our favorite cell is number 7. It's a microglia cell, right? And I think it's, and what is the most frequent cell type in the brain of all these cells? Astrocyte, exactly, yeah, not the neuron. The brain is contaminated by some neurons, but the astrocytes are the most frequent one. Microglia build up to 10% of all the brain cells. And what is the most important cell type in the brain? No, all the cell types are important, They are rare mutations that can affect specific cell types, but we need them all. It's quite clear. So, and and cells like microglia, small cells, astrocytes, astroglia, oligodendrocytes are coined after Rudolf Virchow. He was the first who gave them a name when he was 32, more than 150 years back. And there was a series of lectures he gave in Berlin. It's a general pathology he gave to general practitioners. And in the second lecture, he coined the term of glia. It's from Greek, meaning kid, glue. And it was really a genius idea, to describe the cells in between neurons and to give them the name glia. And at this time point, microglia were not known or not discovered yet, but just all the cells in between neurons. And at this time point, also German was quite popular language in science. So, and here you see it's a picture of astrocytes. And astrocytes, like the name suggests, are star-like cells. Do we have any idea about the functions of astrocytes or any diseases of astrocytes? Well, exactly. What you nicely see here, for example, is a vessel, and you see the processes of the astrocytes going to the vessels, building the blood-brain barrier. And do you know any diseases of astrocytes, although specific diseases of astrocytes we also see them under the microscope. Astrocytes. Astrocytomas, that means brain tumors. Just that you know, these cells can generate tumors. And they are the main population in the brain, you know, and during pathologies, I think this astrocytoma are quite popular brain tumors. We see them in our hospital on the daily basis. Yeah, and you know, here again, vessels and astrocytes giving processes to vessels, and on the other sides to neurons. They are close vicinity to both neurons and vessels, astrocytes. So, and here, oligodendrocytes, another glial cell type, microglia, astroglia, and oligodendrocytes, oligodendroglia. And what is the function of oligodendrocytes for the general audience? Or any diseases of oligodendrocytes?
So the function is myelination, and then you can have brain tumors again.
What do you have, sorry?
Brain tumors.
Brain tumors, yeah, it's the most popular disease of oligodendrocytes. It's also quite common in the US and Europe. It's multiple sclerosis, right? Where you lose oligodendrocytes and you have a demyelination, what we also see in patients, right? So exactly, it's an autoimmune disease, multiple sclerosis. It affects in Germany more than 120,000 patients. In the US, I don't know the numbers. There are also some rare infectious diseases of oligodendrocytes, but as a result, these cells die and the white matter changes because oligodendrocytes are part of the white matter. So, and last but not least, you know, there are several cell types. It's the cell type here, the green one. This is a very flexible cell type, considered as a watchdog or gardener, a garbage man or provider for the brain here in the back of the brain. Our favorite cell is a microglia cell. So microglia cells are immune cells in the brain. This is a big difference to neurons and astrocytes because neurons, astrocytes, oligodendrocytes come from a different lineage during development. And in the developing brain, these immune cells, macrophages called microglia, invade the brain during development. And they are similar to other tissue macrophages. And in all bodies of the brain, we have macrophages. For example, in the liver, they're called Kupfer cells. In the bones, we have osteoclasts. These are all the same family, all macrophages. And also in the lung. And in the brain, these macrophages are called microglia cells. Yeah. So, and for many years, these cells were considered as boring cells, just eating cells, cookie eaters. Yeah. Yeah. But this changed dramatically because what is or became clear in the last couple of years that even if you have no infection during health, these cells are important. Yeah? If you wouldn't have macrophages, you would die off. Yeah? Because the bone wouldn't grow if you have no osteoclasts. Without macrophages, you would become infertile because also the production of sperms depends on macrophages. And even in the brain, there are some rare cases of patients that don't have any functional microglia cells. These brains degenerate over years. Yeah? Without functional microglia cells, no infection at all, our brain would degenerate and people would develop Alzheimer-like symptoms or Parkinson-like symptoms, showing that these cells are important also during steady state. Yeah? It's quite important. So, and again, some history. Ardis Alzheimer already found under the microscope of his patient Auguste Data. These are the famous plaques, the extracellular deposits of amyloid. But he already described here in German at this time point, more than 110 years ago, glial cell. You know, he found around the amyloid plaques in these patients' glial cells. Yeah? And we would consider them nowadays as microglia cells, indicating that these cells can react rapidly and migrate to pathology. So, 1911. And 8 years later, this was the birthday of microglia cells. More than 100 years ago, Rio Ortega described under the microscope for the first time microglia cells. He took sections from patients, did a silver staining, and found these very small, tiny cells. and he calls them, or it was a third element, because at this time point, neurons were already known and astrocytes, but not the third cell type, microglia cells. And his teacher was Cajal, who got the Nobel Prize some years before, but Ortega discovered microglia cells. So the function of these cells was not known at all. So, and for many years, see here, for example, we used H&E staining of the brain. Here you only see the nuclei, the blue dots are the nuclei. You don't see the processes, you don't see the arms. And quite boring. It was in the 80s here, we did H&E staining, where you don't see the processes, and you see the clusters, nodules, accumulation of cells, of hundreds of microglia cells that we found in the brains of HIV patients and also nowadays in COVID-19 patients. Yeah? Just a very unspecific sign of activation. Doesn't look so fancy. And starting in the 90s, where the antibodies were available, and this is an antibody against a microglia-specific markers, where you can appreciate for the first time all the processes here of microglia cells. It's a vessel, and here are some nuclei of other cells, like a neuron here. But here again, microglia form a network across the brain and all brain regions, and in between are neurons. So you just see the nuclei. So, and this was the status for many years, for decades, yeah, until the 90s. And in the 90s, there were new discoveries, new inventions, and microglia research started to explode, really, yeah, 30 years ago. And this is a very important movie, and I will start it in a second. You will see microglia in green. You don't see the neurons in between, but just the microglia highlighted. And what you now nicely see is that these are highly active cells in the living brain during day and night. Yeah? And it's super fascinating. Neurons are fixed, astrocytes are fixed, and the only motile cell type here is microglia cells, continuously extending and retracting the processes. Yeah? And I think if you close your eyes and thinking that inside your brain, microglia cells are massaging your neurons, it's wonderful. Yeah? Even some people can really feel how microglia cells move. It's super cool. Yeah? So to have such kind of cells in the brain. So, and this was 15 years ago. And what we think, this is again a microglia cell in close vicinity to neuron, just one process of a neuron. And what we now appreciate that they have many functions in the normal brain, homeostasis, no disease at all. We need functional microglia cells to keep our brain busy and intact during development, during homeostasis, any kind of learning and memory, where neurons are important and all these processes, so-called synapses, are built. And this is a very dynamic process. Synapses will be built within minutes, and this process of synaptic pruning is modulated by microglia cells. They really touch the processes, they touch the neurons and remove actively the processes. Yeah? CNS adaptation, learning, memory, you need functional microglia cells. So now the big revolution came the last couple of years that many of these diseases, like Alzheimer's, Parkinson's, or multiple sclerosis, are multifactorial cells, meaning many risk factors are important. And they investigated several thousand of these patients. Yeah? And, for Alzheimer's, for example, more than 150 risk genes are known, yeah, that are across the genome, 150 risk genes, and the majority of the risk genes are found on microglia cells. What does it mean for disease? It's not clear, but risk genes can be found on microglia cells. So Alzheimer, Parkinson, and multiple sclerosis. And even for many psychiatric disorders like autism, schizophrenia, where it's known that the people have abnormal synaptic connectivity, and we would like to call them neuropsychiatric disorders or neurodevelopmental disorders. People think that one cause could be abnormal microglia cells that are not properly forming the connections. So, and stroke and other diseases as well, And this is a big hype at the moment, that many brain diseases, schizophrenia, autism, Alzheimer's is most likely at least modulated or initiated by microglia cells. Again, here, take-home messages, many microglia genes or macrophage genes were mapped to brain diseases. So this is for Alzheimer's, Parkinson's. In the Human Genome Project, you can have a look on all the different DNA regions, and you see here so-called genome by dissociation studies. You see risk factors of microglia cells. And this is a recent publication where they investigated almost 50,000 patients suffering from multiple sclerosis and more than 60,000 controls, normal patients. And here they found more than 200 variants on the genomic level. And the majority of them, again, can be found on microglia cells. Yeah? And that's why these immune cells, microglia cells, are major interest, major focus on many companies and also in universities to shape disease. Yeah. Well, and there are very rare patients that are monogenetic, have monogenetic disorders, like one important gene that is important for the survival of cells. CSF receptor, colony-forming receptor, is a survival factor for microglia cells, and there are more than 200 patients now that have an autosomal dominant mutation just in this gene. And this gene is found on microglia cells, and as a consequence, these patients have no functional microglia cell. Yeah? And as a result, in the 50s or 60s, these people get changes in MRI. Here, these white dots are changes in the MRI. And a colleague of mine in Munich did this biopsy, and what he found, severe changes in the brain due to the absence of functional microglia cells. So here, neurofilament, for example, is, you cannot find neurofilament deposits in the brain, just in these patients. The same for APP, indicating that they are dying neurons in the absence of functional microglia cells. Yeah? We would like to call them microgliopathies. It means primary diseases, very rare diseases, due to the absence of functional microglia cells. Yeah? Very rare, but this is in contrast to Alzheimer's and Parkinson's, where many genes are somehow involved in the disease pathogenesis. Right, and this is a kind of more general function of microglia cells, being in contact with neurons. They are considered nowadays as gardeners because they really can prune neuronal trees, the axons and dendrites. They can also be the cleaners, yeah? And alarm clock. They are highly sensitive. And what you have seen in the movie before, they can react within minutes towards any changes of the environment. Very sensitive cells in the garbage man, But they can also be, if they are overactivated, a bad guy, And during disease, because Alzheimer's takes for many years, these cells are living also for years, and microglia cells get overactivated. And they are deposited around the amyloid plaques. That means over the years, they can also become the bad guys. And what is now important for the future, for our research, and I'm really grateful for the GFG to support our research for many years, is really to understand what type of microglia is a good guy and what type of microglia is a bad guy. Because what we would like to have at the end of the day to modulate specific population or specific states of microglia cells. And it started 4 or 5 years ago when new techniques were developed in Israel and the United States. And these were wonderful instruments for the researchers. If we considered microglia as a quite homogeneous population, equally distributed across the brain, all different brain regions. And now we have new instruments to really recognize specific subsets, specific states of microglia cells, yeah? A kind of new microscope. And now it's an age of, we feel sometimes like Christoph Columbus, because we can now measure the immune landscape of the brain for the first time in a high magnification. And these analyses are called, or techniques are called single cell analyses. You can really measure on an individual level cells like microglia cells, RNA level, DNA level or protein level. This gives us wonderful new tools to understand disease on a single cell level. And just to what was done before, you know, all cells were put together and meshed, yeah. If you have your different fruits here, like apples and bananas, everything was for many years meshed together, and we had a kind of average analysis. But since 4 or 5 years, it's now really possible by using single-cell techniques like single-cell sequencing, CyTOF, to analyze all the different cell types, all the different states, and you see all the different parts of the cell here. And the beauty now is that we can really define for the first time disease-specific populations. Yeah? And for the next years to come, I think this is a major challenge to understand specific populations. So, and just to give you an example, what we have done is these are 2 MDs, Roman Zankowski, Uri Zachewski in our institute, and Takahiro Masuda, a postdoc. And in the last years, we are really the first who could decipher in the human brain microglia states during disease, and Marta Yoda was also a postdoc. And, you know, she took human brain, part of the human brain, used single-cell techniques. Cells were really individually sorted here and sequenced, and they could really analyze.
Can you go back?
Sorry, yeah.
I missed that last part.
Yeah, sorry. Analyze and analyze different cell states on a single cell level, right? And here, for example, this is the new landscape of the brain we can establish. It means each dot is one cell, yeah? And gives information about thousands of genes. Before it was really one big cloud, yeah? But now we get a much better resolution, can really define individual states of cell. Each dot is one cell. For example, here we have 9 different clusters. And now for the first time, and this is something for the future, we can really define what part or what cells, what microglia cells are the gardeners and the important cell types that we would like to keep. What population is alarm clock, and we also would need in the human brain the cell types still be present. And what, for example, is the bad part of the microglia cells, since we would now like to understand also with the help of the DFG support to understand what state, what population during disease in the human brain is a bad guy. And in future research, we would like to analyze this more in depth and also hopefully to find a way to interfere with this population. You know, it's really very new landscape we have. So, and you know, it's, you know, idea would be, and this is also for other researchers in cancer research and neurodegeneration, neuroinflammation, and also here in New York, I realized that these single cell techniques, also for brain diseases, it's the future direction. We would like to analyze disease-specific states and populations that are specific for Alzheimer's, for multiple sclerosis, with different technologies. Here, these are listed high-resolution microscopy, or these different techniques like sequencing or CyTOF on protein level. So it would allow us to diagnose the brain totally, completely, or new way, and to identify, as I said, new specific populations that can hopefully be targeted, and that we can redefine diseases in a new way, and to have new targets for disease and also new screening methods. So it's a future, and here I think, yeah, that's it, because I would like to summarize, you know. I hope that you got the main message of this talk. You know, the most important cell type is microglia cell, yeah? And they are living for years, and we and others have shown that they really stay in your brain for years and decades, yeah? They have no exchange with blood cells. That's why you have to treat them carefully, be nice to your microglia cells. And it's always a team effort. It's not just me, many people here. And these are, you know, these are so-called Bollenhut, because Freiburg is localized close to the Black Forest, so we very typical here. So, and if you think that these people are totally relaxed in the lab just playing around with red balls, it's not true. They are really here interested in the most exciting immune cell type in the brain called microglia cells. And special thanks to DFG, you know, it's not, I'm not saying this because I got invited by the DFG now. Over the last couple of years, really received millions of euros for our research, and that's why we are so successful over the couple of years. I'm really grateful for the DFG. Very important support for many years. And the last slide, you know, we are placed in Freiburg, in the south of Germany. And it's a wonderful place because it's not New York, it's a small city. Obviously, we have wonderful buildings like the medieval cathedral here. And if you look closer to this cathedral, you see funny sculptures here, reflecting the great sense of humor of these people. Yeah. And people like to live in this city. We have big rivers inside the city center. Here it's a big river. It's called Bächle. The kids really like it in summer. It's because it's hot in summer times. Kids like to play around. We have research institutes like the Max Planck Institute in Freiburg that is also quite popular. And if you are lucky and are walking throughout the city, you find popular people. Like for the Germans, he was very important. He's the national coach of the soccer team, yeah. But retired after 14 years now. But exactly, and why it's important for us, it's a neurocenter where we are localized here and doing our research and diagnostics. Thank you very much indeed.
Yeah, Marco, thank you very much for that fantastic talk. A lot of insight. I'm sure there are questions in the audience. Who is? Yeah.
I was just wondering how large these cells are.
Well, in terms of diameter, they are, well, they are tiny cells, and with extension they are 10, 15 micrometers small, yeah, quite small, and the whole brain is really packed with billions of cells. Astrocytes are much larger. And as I said, microglia are really, as the name suggests, small cells, 10, 15 micrometers. It's also a disadvantage because if you want to isolate these cells for molecular analysis, we have to sort many, many cells to get information. Yeah. They have very few RNA, very few protein compared to neurons. Neurons are big, huge cells. Yeah, small but beautiful. Yeah.
Next person back here.
Yeah, thank you. Thanks very much for the talk. I work in public health, and I don't really get a chance to, dig into this kind of research. So it's nice to be able to shift my brain. I was not aware that we were at the point where we could actually do this single cell sort of sequencing and analysis. So I think that's tremendous. But I wondered, as you were grouping the cells there, and we talked about all of them, and of course, the bad guy, and it just makes me wonder, like, you also have to, I guess, try to, or do we understand why these groups do the things that they do? So when I think about bad guy, if they're in there and they're doing that, and they're not, it's not an outside force that's like a virus or something that's making them do it. You think usually they're doing one thing at the expense. One thing is at the expense to protect something else.
Yes.
But maybe that's not.
Yeah, well, it's a very important question. And for example, we are also using this 2.5 million from the GFG to cross inside to look in the brain across different pathologies. We have more than 30 pathologies, but we are now looking for different microglia phenotypes, to really do the single cell sequencing in the brain of Alzheimer patients compared to multiple sclerosis, stroke, brain tumors, to really understand what is specific for the microglia behavior in given conditions, also in different brain regions and across development of the brain. There are so many open questions, and so far we only see kind of snapshot, a fingerprint of microglia cells. We do not know the function at the moment, but it's getting more and more complicated. But by collecting different information, we hope that at one day we know more.
Maybe I'm going to give the microphone to the person in the back because otherwise.
Thank you. Hi, good evening. It was a fascinating talk. Thank you so much. My question is probably a little not so scientific. But I'd be very interested to know if our thoughts or our attitudes or Lebenseinstellung, does that have an effect on the brain and its development and the cells and what you spoke about this evening?
Well, yes and no. I didn't show data from a study we obtained a couple of years ago. We were the first who basically discovered a connection of host microbiota, so that's sitting on the skin and gut. Yeah, because for this study we used germ-free mice 5 years back, and it was also confirmed in patients, in humans. Yeah, that what you eat is important for the composition of the gut flora. In this gut flora, bacteria, for example, E. coli or other bacteria, secrete factors that get access to the brain and modulates the immune system in the brain. You know, it's quite important because if you take antibiotics, for example, you remove your bacteria. This would also have major influence of the microglia, as we have shown before. They look totally different. They respond differently to different diseases of the brain, and it's a very important communication of the gut and brain. You know, our research opened many ways in this direction.
I have a very provocative question
Yeah.
in that same direction. Because it was really groundbreaking study where I think you for the first time showed how the diversity
Yeah.
of the microbiota in the gut actually is really important so that the brain functions normally. Now, we've seen a lot of increase in some of these neurodegenerative diseases. We also see a lot of increase in autoimmune diseases, others. And many of them are correlated to the industrialized world. So people that live in a not so clean environment where probably there's much more diversity of bacteria in the gut, and maybe even more challenges to the immune system as well. Could that be a reason why we have an increased incidence of Alzheimer's and those kind of diseases that are just due to the fact that our gut microbiota are different now than they were in the past?
For Alzheimer's, it's quite clear we have more cases because the life expectancy is increased. You know, and if you get older, the likelihood is also much higher to get Alzheimer's. Because 150 years ago, the mean life expectancy was 45 years. Now it's much more, and I think the brain or the immune system in the brain, like microglia cells, they can only cope with amyloid for some years, but not for decades. You know, and for sure the environment is important, what you eat is important, and also the immune system is important. I think it's very complex interaction in many ways.
I think very interesting. More questions?
This is more a biographical question. Was there one experience, or was it different experiences in high school and college? What got you into this field?
Well,
the cookie monsters.
Yeah, the cookie monsters, you know. Well, I was already interested in the brain when I studied medicine, you know. And after finishing my medical school, I was looking what can I do in terms of neuroscience research, you know. And in Berlin, at this time point, late 90s, there was a Max Delbrück Center in the north of Berlin, and there was one group working on these boring cells, microglia cells. Nobody in neuroscience was caring about the cells because all neuroscientists were working on neurons, very few on astrocytes, but nobody on microglia cells because these were, at this time point, considered to be very boring cookie eaters, you know. And I'm grateful that I started my career 25 years back just on these boring cells that were now in the focus and really center stage in neuroscience research, you know. Yeah, just by incident. Yeah.
Pays off sometimes to go.
Yeah, I also have a question that is less scientific because I'm also a studies person. But you have this fantastic team.
Yeah.
And you do wonderful, beautiful actually work that has fantastic images and everything. How do you choreograph your lab? So if I walk into your lab in the morning, so what happens there? How do you start? How do you go through the day? What does everybody do?
No, they behave like soldiers. No, just kidding. Yeah. No, I'm East German. I grew up in East Germany. That's why. No, you know, well, for example, one important experience was in Zurich, Switzerland. You know, when I was working around 2000 in the hospital in Zurich together with other very famous scientists like Adriano Aguzzi working on prions, and Charles Weismann, who discovered type 1 interferons, and Rolf Zinkernagel, who won the Nobel Prize for Immunology. All these people were together at this time point in Zurich and also working together. So it means very international teams, with people coming from Asia, from Northern America, from Europe, and also working during day and nights, including weekends, you know. It was really terrible times, you know. And also, you know, but then I learned that science is very, well, yeah, it's driven by passion for sure, but these people really like to do science. They really like to discuss science and to bring new ideas and to develop these ideas. You know, it's very international, and now I'm the privileged situation that people are coming to my lab, bringing their own money. Yeah, it's really cool that they really want to study in our institute, and it's a very privileged situation. So, yeah.
It's all about the academic freedom, no?
Yeah, exactly.
Hi, my question is like a little bit too technical, so if this is not appropriate, I can ask you later. But I was wondering, so like in ALS or
Yeah.
like other neurodegenerative diseases, there's an overexpression of certain factors that we think might be responsible, like TP43 or SOD1,
Mhm.
things like that. And then there are some papers that talk about if you can, if you're able to create that suppression, or like in animal models also, they see an increase in like microglia. And like I was wondering, I was more on the translation like industry side of things. I was wondering your research, like what could be certain pathways or implications to think about in terms of making it interventional, therapeutic, or translational? Like how could we use microglia in the direction of, and it doesn't have to be specific, just ideas.
Sure. Now, I also wrote 2 years back in Science, in Viewpoint, addressing this question, you know. And usually microglia are good guys during neurodegeneration, but over the years, if they are overactivated, they get exhausted and not able to remove proteins, for example, like in ALS and so, yeah. And idea would really to find strategies to replace these cells, to give them back power to phagocytose, to remove debris. And this is something for the future. And we would like to understand the exhausted microglia cells and to find ways to replace them with new microglia cells that are powerful and also remaining the good guys, yeah, that are important for survival of the cells, you know. It's a very complicated situation, but I think it's aim for the future.
But this is another interesting point I think that you also discovered very early on, is that these microglia cells, they migrate to the brain very early on in the embryonic development, and then they're no longer being formed, so they basically stay around, or do they multiply inside the brain?
Well, they are still plastics, and there are also ways to deplete the majority of microglia cells. You can get rid of at least a mouse brain, and they also, since they are plastics, they come back. They fill the niche again, but this is the beauty, is that they are plastic cells, and at least in the future we hope that we can modulate them. Yeah.
But they don't cross the blood-brain barrier.
No, not exactly. And they live for years or decades inside the brain, very slowly proliferating.
Because this was also interesting, because most other macrophages are produced by the bone marrow normally, and they are continuously regenerated in the course of your life.
Yeah.
So this, I think, is the only population that
this
formed very early and then kind of
this discovery we made together with the scientists from the Mount Sinai Hospital, Frederick Geisman, from French researchers, that most tissue macrophages are derived from the bone marrow, as you said, but some seed already prior to disease, prior to birth, like in the brain. And so, brain is very special in many aspects.
More questions from the audience?
Yes, regarding the international exchange, as I understand, Freiburg is one of the main focal centers for research in microglia. How many other centers are there, and what is your interaction? How important is this exchange in the promoting of your work?
Yeah, well, microglia are really, I think, in the border between neuroscience and immunology. Yeah? And that's why we have many cooperation partners working on either just neuroscience or just immunology. And, well, science is very international and it's a big network, you know. That's why we have different partners in Europe and different partners also in the States, you know. It's very international, I think. You always need the leaders in the field, yeah. For example, for single-cell RNA-seq, We have collaborating partners at the Weizmann Institute. We are really pioneers of these techniques, and we are using these techniques to get new ideas and new data. Science is really very international. That's why I think I hate Zoom meetings in the meantime. Yeah, I prefer to meet in person and so. Yeah.
What's the unique selling proposition of Freiburg?
Selling?
Unique selling proposition.
Oh. That you don't need a Porsche.
Basically, I have a bike. Yeah. Green city. It's a green city.
That's why the Leibniz Prize for somebody from Freiburg.
Exactly. Yeah. And the soccer team is very special, and so SC Freiburg, very special. Yeah.
No, I think the fantastic part is actually climate. You know, what most people don't know that when it's grey in Germany, like in November,
exactly.
You just have to walk up a little bit the hill, and then you get this fantastic, beautiful sun.
Exactly.
Over
The Black Forest. Yeah.
There's always an inversion.
Exactly.
Which I really like when I was studying there, because you could just escape the grey, dark winter weather in Germany and go somewhere else. And that's really unique to Freiburg.
And very close to Switzerland. If you want to go in 30 minutes to Switzerland or to France, it's very.
When you study like and you skip your lectures and they go skiing.
Exactly. Exactly.
There's a lot of really, I think, very nice arguments.
There's a question in the back.
Yeah, I would just add to that. I lived in Würzburg in the 1980s, and I can just say that that whole southern Germany is worth visiting. I mean, Berlin is great. Frankfurt is nice.
But Freiburg is the best place to live.
Absolutely. But my question was, so I wonder, this is very exciting kinds of research that can lead to a lot of things I think people would be really interested in. I know that when you are a researcher, your main way of communicating out is publications, and I Googled you really quickly, so I know you're a prolific writer, and you're on a lot of articles, but I wonder how else you communicate these out. Like I noticed you had a few things on YouTube, and I didn't know if there are other ways to kind of get people that aren't really reading, peer-reviewed research to get excited and interested in what you're doing.
Yes, well, it's a good question, Basically, my main interest is also to have a good education of students, and also for the new researchers, the new generation of researchers is important, that are trained in the best ways, they get the ideas, and that really drives research, I think. I'm really supporting the young researchers, and we have many young MDs and also PhD students, and we are supporting them, females especially, and this is something for the future.
But it is something really important, and maybe I can just throw in something what we are doing in this respect, because I have seen the same issue. Of course, scientists like you and what we're doing is very, very fundamental science. It's very hard sometimes to explain it to the general public. But there is something that I think all those kind of scientists have in common, that's curiosity. And so what we basically started to realize that most of this fundamental science that Max Planck does, that you do in your research, is curiosity-driven discovery. And I think if you understand that this is what drives him and all his colleagues in the lab to work 15, 16, 17 hours a day and stand up and still come the next morning again. This is a really important factor. So what we've done, we've launched a lecture series that's called Curiosity Meets Discovery from Max Planck, Florida. But that covers actually a lot of the science of the Max Planck Society in Germany is being streamed in English, and it's being trying to kind of present the science in a publicly accessible format, so that normal, educated, science-interested public can actually kind of learn what's really going on behind the scenes, so that they have an opportunity like today to get a little bit better insight in this. So if you just Google Curiosity Meets Discovery, you'll probably find access to those lecture series. We just started actually, had one today of Rainer Genzel, who is a Nobel laureate of the Max Planck Society about black holes. But it's covering the entire spectrum of fundamental research. But I think it's very important that the scientists have opportunities to communicate with the public, but of course sometimes it's also hard because we're doing so much specialized stuff that it's not always easy to do that. More questions? Well, I have tons of questions.
For the break,
That's just because I'm an immunologist. I kind of gets me back into my PhD times. The one thing that I'm really still fascinated about is this diversity story that you founded. basically if a lot of diversity is needed in the body to generate homeostasis, and as soon as you basically cut on those diversity, things start to go wrong.
Yes.
And so, do you really think that with this understanding this, we can actually repopulate, especially in disease cases like autism or so, where we know that, that it's because of a limitation things don't work, that you can kind of regenerate the normal state?
Of specific cells? Or
specific cells, or even like, I don't know, regenerate, as you said, it's microglia, probably the pruning of synapses. So in autism, I think
well, it's always a combination of internal factors like genetic factors and environment, you know, that shapes a cell identity at the end of the day, you know. It's both factors are important to a different extent, and we really have to also realize the input of the environment, you know. The neighboring cells and external factors, many factors play a role. Not easy to answer.
Well, if there's no more questions by the audience, I think then...
There was one question, yeah.
I have a question related to Alzheimer.
Yeah.
So you mentioned the relationship between microglia and amyloid is important. I was wondering how important you consider the relationship between tau and microglia.
Yeah. Well, exactly. Amyloid, A-beta 40 to 41, is found extracellularly, and as a cleavage product of APP, whereas tau is inside the neuron, right? This abnormal phosphorylated tau you find in-cell microglia, inside the neuron, and only if a neuron dies, you see tau positive outside the cell, you know. So, and even for Alzheimer's, it's not clear what comes first, you know, whether amyloid comes first and tau follows, what the majority of people think, or vice versa, you know. What is clear, and there were also studies not by us, but by other people, that there are specific microglia states or clusters in the human brain that are specific for either a tau type of Alzheimer's or amyloid type. So that means the cells are able to react in a different way to different pathologies like either tau or amyloid, you know. What does it mean in terms of function or therapeutical approaches? We don't know yet. But they can smell the different pathologies. Let's put it this way.
One more.
Curiosity.
Hi, so we know that there are these different reactive states of microglia, right? So if you have like a general inflammatory insult to the body, even though that should theoretically cause like the same reactive state throughout the brain, you will see different reactive states of microglia afterwards. Do you think once a microglia becomes reactive in a particular way, it has to return to homeostasis to go to a different state, or it can transition?
Yeah, it's a very good question. You know, in humans we cannot prove it. Yeah, but we develop strategies to prove it in mouse. You know, genetic tools to follow the fate of a microglia cell during pathology and afterwards. If an insult is over, we have a resolution phase. We would like to understand whether a microglia cell can return to a normal state or not. This is something for the future we are interested in, definitely.
I have one in between.
No.
So in the picture that you showed on the where you can kind of classify these different types, can you also do this in situ? So can you actually see in which part of the brain something is going on already?
Yes. I think if you look for the spatial information, spatial analysis on RNA or protein level, you can do it. But it's always limited to a number of genes or limited number of proteins, but you can do it. And this is what we're also doing.
Because I think the position is always important.
Yes.
So in Alzheimer's disease, microglia is known to be responsible for phagocytosis and the evaluation of A-beta. But a few months ago, this paper came out from the Planck lab showing that microglia can act as trash compactors, and instead of degrading A-beta, kind of like making dense core plugs to make it not available. I wonder if you have any comments on that.
What study was it?
It's some receptors regulating microglia activation.
Which receptors?
It's Planck lab. It was in Nature Immunology.
Okay.
So they suggest instead of degrading A-beta, microglia can...
These are the multi-K receptors, right? This family of...
sound receptors.
Yeah, exactly. I think we discussed this paper during a journal club. Yeah, it was one special model, APP-PS1 model, right?
It was APP-PS1.
Yeah. But I don't have all the details in my mind, but it's always, you have to look what state of disease, what model, and so it's always a complicated situation.
I think it's a very interesting topic. You see that everybody is curious to learn more and to hear more from you. So I nevertheless think that there's time for talking in a little bit more relaxed environment. And before we do that, Martha will kind of say a few words to you, and then we, I think, go over for this.
Sounds good, yes. I'll just give a few closing remarks. So I'm Marta Miller. I'm the representative of the University of Freiburg Liaison Office.
Freiburg.
Yes. So, of course, I'd like to say thank you to Dr. Haury and Dr. Prinz for this very insightful and wonderful conversation. And I'm not just saying that because of your connections with Freiburg. I would like to say thank you to all of the teams who helped organize this event, from the DWIH and the DFG, and of course to the German Consulate General for your ongoing support of all of our events here. And last, of course, I must thank 1014 for your wonderful hospitality. Thank you, of course, for having us here in this beautiful space tonight. With that, I would just invite you all to continue this conversation in a more casual space at the reception outside. So, thank you very much.